discuss about the secondary growth in case of plants. Before going directly on to the secondary growth of plants, first we will discuss the basic things, what are different types of growth and what is growth curve, how growth is affected by the different stages of life cycle in case of plants. As we will define the growth, it's an increase in size. It's an increase in size, volume or mass. It's an irreversible and the permanent process. Directly or indirectly, it will lead to increase in the dry weight of plants. So if we'll talk about the types of growth, we are having primary and secondary growth, unlimited growth, limited growth, vegetative and reproductive growth. So if I will talk about the primary and the secondary growth, in case of primary growth, the shoot apical meristem and the root apical meristem increase in the length of the plant via the mitotic division of meristematic cells. These root and shoot apical meristem or the root apical meristem are present at the apex of the plant. So this was about the primary. If we talk about the secondary growth meristems, obviously the growth is due to the activity of the meristematic cells. The secondary meristem increases the diameter of plant, and when the plant increases in its diameter is called as the secondary growth. So if we will talk about the unlimited growth, so root and shoot are continuously growing till starting from the germination stage to the death throughout the lifespan of the plants. This is also called as indeterminate type of growth. Again, I will repeat, indeterminate type of growth. Second is the limited growth. Like when there is a formation of leaves, fruits or flowers, after attaining a certain size, they stop their growing. So this is called a limited type of growth, or we can call it as determinate type of growth. And the last is vegetative and the reproductive growth. In case of vegetative growth, or we can call it as earlier growth of plant, when plants are able to produce leaves, stem and branches. In case of reproductive growth, this is obviously after the vegetative growth, plants produce flowers which form the reproductive part of the plant, and this is called the reproductive growth or the reproductive phase of the plant. So second, if we will talk about the growth curve in case of plants, we have seen the growth curve in case of plants is having three different phases. Like we are having lag phase, we are having log phase, we are having another the stationary phase. The lag phase is the initial stage of growth where the rate of plant growth is very slow. As it is lag phase, we can call this as a preparatory phase as well. Then in case of log phase, again we can call it as exponential phase. It is where the plant shows the rapid growth and is maximum during the lifespan of the plant. We will call this as another the stationary phase. Here the growth starts decreasing and finally it stops. It comes to a stationary phase. So as you can see in the diagram, if you will see the typical diagram of the plant, we are getting the sigmoid type of growth or S-shaped growth curve. We can measure growth in terms of increase in length in case of root and stem, increase in area or volume. This can be achieved by in case of leaves and fruit, increase in number of cells in case of plants, algae and bacteria. If we talk about the how growth starts, so first is the cell formation, means cell division. Then the second part of this is the cell enlargement where cells grow. Then after its growth, cell enters into the cell differentiation. So during growth, meristematic cells are able to pass through these different three stages. First is the cell formation phase, or we can call it as a formative phase. Second is the cell enlargement phase or phase of elongation. And the third is the cell differentiation phase, or we can call it as a phase of maturation. Cell formation phase. So cell has to form. So obviously the meristematic cells will divide to form the new cells. This division is restricted at the apex, either in case of root or either in case of shoots. So the newly formed cells which are formed by the cell division are having thin cell wall, but they are having dense protoplasm. Obviously, the daughter cells have the same number of chromosomes and the same genetic constant as we can see their mother cells. So this was cell formation phase. Now the second is the cell enlargement, where cells enlarge. These cells, when there is after cell formation, these cells absorb water by osmosis process, which result in the increase in turgidity, which results in the increase in the turgidity. If there is increase in the turgidity, then cell expansion will take place. So the cell enlarge in all directions. जब पानी अंदर आ जाता है due to the osmosis, इस osmosis की वजह से क्या हो जाता है, जो सेल की turgidity है वो increase हो जाती है. तो it's not the turgidity increase हो रही है तो हर एक तरफ से in all directions. लेकिन फिर लास्ट में जब स्पेसिफिकली फिर ऐसा देखा गया इन केस ऑफ प्लांट्स, दिस डायरेक्शन और द एनलार्जमेंट इज स्पेसिफिक और कन्फाइंड टू अ स्पेसिफिक डायरेक्शन एज वेल. सो सेल डिवाइड हो गई, सेल एनलार्ज हो गई. देन द थर्ड पार्ट इज द सेल डिफरेंशिएशन. This is not happening where cell is forming, this is happening below the cell formation. This is called also the zone of elongation. तो cells ने जब क्या किया, attain की एक maturity size, then there is a gradual increase in the thickness of the cell wall. क्योंकि cell permanent phase में जाएगी, meristematic cells to जो है, we have already these things. Cells will enter into the permanent phase, permanent types of tissues, where they will attain the thickness of the cell wall. There will be the gradual change in the structure and physiological changes that will depend on the location of the plants. The time interval from when the cell is formed or cell formation phase up to the cell enters into the differentiation phase, the whole period is called as the grand period of growth. This is called the grand period of growth. As you can see in the above diagram, that the apical meristem can be divided into three main parts, which are considered as the primary meristem in case of plant. Like first is the formation of protoderm, which is again the primary meristem. Second is the ground tissue, which is again the primary meristem, and third is the procambium. So from primary meristem there is the formation of the primary tissue. From protoderm we are getting the epidermis, and the ground meristem we will get the primary tissue in the form of ground tissue, pith and cortex. Then in case of procambium we are getting primary phloem and xylem. Then again there is the lateral meristem formation, as we can say that the ground meristem is converted to primary tissue, ground tissue, that is pith and cortex. Then this primary tissue is converted to lateral meristem, that gives rise to the cambium. Then in case of procambium, the lateral meristem is the vascular cambium, which is directly coming from the procambium. Then again there is the formation of the secondary tissues, like in case of ground meristem, there is the formation of cork cambium as a lateral meristem. Then from the cork cambium there is a formation of cork and phylloderm. So in case of procambium, directly there is a formation of vascular cambium. Then from vascular cambium, the secondary tissue arises in the form of secondary phloem and the secondary xylem. So if we will talk about the secondary xylem makes the wood of the plant, and the secondary phloem, phylloderm and the cork makes the bark of the plant. So now if we will talk about the plant development, we can characterize plant development by the increase in the girth. This increase occurs when primary growth of the plant ceases. Means जब apical meristem और shoot apical meristem या root apical meristem growth cease करता है, that time secondary growth is the result of that time the secondary growth occurs. But this secondary growth is the result of vascular cambium and the cork cambium, which is present in the plants, and the collectively vascular cambium plus the cork cambium is called as the lateral meristem. At each growing stage, there is the reinitiation of radial growth from these lateral meristem for successive years, which result in the increase in the circumference or the diameter of the plants up to several meters. Means पूरे साल के साल ग्रोथ नहीं होती है, जब ग्रोइंग सीजन होता है तब जाके सिर्फ ग्रोथ हो जाती है. Vascular cambium forms the wood, and the activity of the lateral meristem is recorded within the wood by the formation of annual rings. एक साल में जितनी ग्रोथ होगी, अगर फेवरेबल कंडीशंस होंगे तब अच्छी खासी ग्रोथ होगी, और अगर अनफेवरेबल कंडीशंस होंगे तब अच्छी खासी ग्रोथ नहीं होगी. How we will be able to determine the growth? We will be able to observe the annual rings. If these are dark color rings, then it means the growth is high. If we will see the light color bands or light color rings in the girth of the plants, it means कि unfavorable conditions में ये growth हो गई है. When there are, as we can see now, when there are favorable environmental conditions, that time growth rings are thick formed, and under poor environmental conditions, growth rings are thin formed. So how the initiation of secondary thickness occurs? Cambium appears to initiate growth when primary growth ceases. So primary vascular bundle differentiate from the cells of the procambium. This procambium retains ability to initiate lateral meristematic activity. Procambium में ही ability होती है, वो क्या करती है, initiate करते हैं lateral meristem activity को. Cambial activity is normally first initiated within the vascular bundle. So if we have to discuss about the secondary growth, first of all we have to discuss how this cambium ring is formed. This cambium ring is involved in the formation of secondary xylem and secondary phloem. As you can see in the diagram, how the formation of cambium ring occurs. Vascular bundles are arranged in a circle in case of dicots. In case of monocots, they are haphazardly in the plants. But if we have to differentiate between monocot and the zygote stem, we have to see if the vascular bundles are arranged in a circle. That means the given explant is the dicot. Xylem is always towards the pith and the phloem is always towards the periphery. And in between them, if we will see the xylem and the phloem, in between them there is the presence of the cambium tissue. And this type of arrangement of vascular bundle is called as open vascular bundle. The cambium tissue, which is formed between the xylem and phloem, is called इंट्राफेसिकुलर कैम्बियम. इंट्रा, ओके, जो जाइलम और फ्लोएम के बीच में जो कैम्बियम टिश्यू है उसको बोलते हैं इंट्राफेसिकुलर कैम्बियम. एंड द स्पेस बिटवीन द टू कैम्बियम सेल्स इज कॉल्ड द मेडुलरी रेज. और जो स्पेस है एक वेस्कुलर बंडल से दूसरे वेस्कुलर बंडल तक, उसको क्या बोलते हैं? मेडुलरी रेज. These medullary are parenchyma cells. The line between the two vascular bundles in the medullary rays, that is the parenchyma cells, will get converted into the meristematic cells. And these meristematic cells, as you can see, these are called, as you can see the diagram, these are called as interfascicular parenchyma. These are called as interfascicular parenchyma. That is the cells of the medullary rays which are adjoining to these intrafascicular cambium becomes the meristematic cells to form the interfascicular cambium. These both types of cambium, that is intrafascicular cambium and the interfascicular cambium, join together to form a complete ring. As you can see on the right hand side, there is the complete formation of the cambium ring. From these cambium cells we are able to get the secondary phloem and the secondary xylem. These secondary phloem and the secondary xylem are involved in the secondary growth of the plants. This cambium cells become active and is involved in the formation of new cells towards inwards as well as outward. जब ये कैम्बियम सेल बनती है फिर इसमें सेल फॉर्मेशन हो जाती है अंदर की तरफ प्लांट और बाहर की तरफ. इस रिंग से अंदर की तरफ सेल्स बनती है और बाहर की तरफ सेल्स बनती है. These cells which are formed outwards or towards outer side, these cells which are formed towards outer side gets differentiated into the secondary phloem, and those cells which are formed towards inner side gets differentiated into the secondary xylem. Means secondary phloem बाहर की तरफ बनता है, secondary xylem अंदर की तरफ बनता है. It has been observed that the cambium is more active on inner side than the outer side. पहले cambium की हमेशा देखा गया कि ज़्यादा से ज़्यादा सेल्स अंदर की तरफ बनती है. This results in the more secondary formation of secondary xylem than the secondary phloem. This जो ये हमारा secondary xylem is thus making the main bulk of the plant body. The secondary xylem, which is forms the tracheids and the vessel elements inside, and the secondary phloem are involved in the formation of sieve elements and campanian cells which are towards the outside of the plant. The thickening of the system is due to the formation of only secondary phloem and the secondary xylem plus the action of cork cambium. This cork cambium forms the tough outer layer of the stem. जो हमारे cork cambium है ये बाहर की तरफ होता है and ये हमारा tough part बनाता है किसका? stem का. The cells of the secondary xylem which is towards the inside contain lignin which provide hardness and strength to the plant trunk. If we will talk about the woody plants, the cork cambium is the outermost lateral meristem. It produces cork cambium bark, as we discussed earlier. This bark contains a waxy substance this bark is having some waxy substance that is called as suberin that can repel water. The bark protects the plant against the physical damage. It protects the plant from physical damage. Second is it doesn't allow pathogen to come inside the plant, and it also helps in the water loss. It also helps to reduce in the water loss. The core cambium also produce a layer of cells which are called as phylloderm, which grow inwards from the cambium. The core cambium and the cork cells and the phylloderm are collectively termed as periderm. We have already discussed about the periderm in our previous lecture. The periderm substitutes for epidermis in mature plants. In some plants, the periderm has many openings. These are called as lenticels, which allow the interior cell to exchange gases with the outside atmosphere. This supplies the oxygen to the living and metabolically active cells of the cortex, xylem and phloem. This is the how secondary growth are occurring inside the plants and which cells are responsible for the formation of secondary xylem and the secondary phloem. Now, if I will talk about what is the direction of the growth inside the vascular bundles, how cells are getting differentiated into xylem and phloem. As you can see in the diagram, this is the flow chart. This is how the direction of growth occurs. First, on your left hand side, you will be able to see the cells of vascular cambium, which initiate the secondary growth. So first step is the division. So this vascular cambium cells divide first, form another vascular cambium cells, that is the CC. One C is dividing into the 2C. Then this one C will remain as such because they have to go for further activity. So the second C is able to get differentiated into the xylem, which is towards the inside. Now the upper C again gets differentiated to form the CC, and the xylem which is formed in after the first division of cell cycle forms the X1. So the X1 is the first cell, second will be the C. Now the upper C will get converted into the first phloem, P1. Then again, this was the one complete cycle till here. Then again this cambium cells will get differentiated. So one C will form the another C. But on its outer side, the P1 will be there, that is the phloem, and towards inside it will be X1. Now again this another C will form the X2, that is xylem 2. It means X1, X2, then cambium activity, cambium cells, then the P1. Then again this C will get converted into the form two C's, two cells, that is we will be having X1, X2, CC and P1. Now again the second C will get converted into the P2 phloem. So we will be having the arrangement of cells like this: X1, X2, C, P2, P1. Then again the third division of cell cycle will start, and like this the plants are able to have the rings, plus this is how the secondary growth is occurring inside the plants. So if we will talk about the growth rings, so we will take the cross section of the stem in case of woody plants. There we will find the growth rings that are actually one ring is the single growth increase in the single growth period. मतलब एक रिंग उतनी बड़ी बनेगी जितना एक साल में उसकी ग्रोथ हुई है. If we will talk about the plants which are present in the temperate regions, their growth is actually 1 year. Their growth is actually 1 year. So we will be able to see their growth ring, or we can call their one growth ring as a annual ring. But in case of tropical region plants, growth ring may not be annual. Growth ring may not be annual. There may be more than one ring, which we can call as second or false ring, and may be deposited during single year. We can see two different types of growth rings. We call them as, like, if the growth ring is produced in the early spring, we call this a spring or early wood. Or if it is produced later, we call these as summer or late wood. Sometimes it has been observed that the growth is terminated by the development of thick-walled fibers or by the parenchyma cells. In case of the plants which are from cold climate or temperate regions, the age of the tree may be determined by counting simply the number of annual rings which are present in the base of their trunk. If their trunk is hollow, मतलब we don't have the मतलब like strong trunk, it's a hollow, means ये खाली है अंदर से, then we can check these growth rings at the base of their large roots. उनको root को cut करेंगे, there we will be able to observe the growth of the plant by counting the number of rings there. Even these annual rings we are using for even ancient wood structures. By counting these rings, we are able to determine the growth of those ancient dating wood structures. Mostly this technique was developed or used in American journals, or we can say American Indians in the dry southwestern region of the USA. किसने यूज किया? जो अमेरिकन इंडियंस है in the dry southwestern region of the USA. Even they have confirmed from these rings, they have observed sometimes there is a fluctuation of ring width, which they, from this ring with confirmation, they are able to get the information about the ancient climate. They are, if, as we have discussed earlier, that if there is a good growth, favorable climatic conditions are there, then the growth ring will be quite big, or it will be मतलब ज़्यादा लेंथ में होगी. अगर एक क्लाइमेट कंडीशन नहीं है तो हल्की सी ग्रोथ रिंग हो सकती है. So this type of study is also used for dating ancient modern structures as well.